Excellent. Well, welcome everyone. Ani, bonjour, and bienvenue tout le monde, spécialement nos membres de FishCAST qui viennent de Québec. Welcome to the final seminar of our FishCAST Fall Seminar Series. You can actually catch up on our previously recorded seminars on our YouTube channel, and Kendra, who's FishCAST's program coordinator extraordinaire, she will be posting the link in the chat, Kendra, if you don't mind. So you can catch up on, we actually started our seminar series this year in May. For those of you who don't know me, my name is Dr. Christina Semeniuk, and I am the director of FishCAST, which is an NSERC Create funded program. And we're designed to train the next generation of leading experts in the conservation and management of Canada's freshwater fishes. And you can learn more about us at fishcast.ca. Before I introduce our speaker today, I just wanted to go over a couple of housekeeping rules. And that is, if you don't mind, just double check that your mics are muted. Your videos can be off just to help with bandwidth. Even Dr. Zielinski said that she'll keep her camera off because she's talking to us from a remote cabin up north in Ontario. But afterwards, please feel free to post any questions that you have in the chat, or you can turn on your audio and your video to do it as well. One of our members, Dominic, already made sure that our chat does work for guests, so please take advantage of it if you will. So to begin with, I'm so delighted to present to you our speaker today, Dr. Barbara Zielinski, who is a member of our FishCAST academic team. And Dr. Zielinski investigates chemoreception in wild fish. Her NSERC-funded program explores the diversity and convergence in the olfactory system of fish. But Dr. Zielinski not only is an expert in the pure sciences aspect of this research discipline, but she also has a really long-standing collaboration with the Great Lakes Fishery Commission to study the olfaction, the chemosensory system, and now the taste system of our invasive sea lamprey in the Great Lakes. To give you a bit of background of Dr. Zielinski's academic history, well, she obtained her bachelor's in science as well as her master's of science in biology from the University of Waterloo. She then went to the University of Manitoba, where she did her PhD in the lab of Toshiaki Hara in neurophysiology on fish chemoreception. And this took place at the DFO's Freshwater Institute. For Dr. Zielinski's postgraduate research, she went first to Wayne State University in the Anatomy and Cell Biology department, and her research there was on the secretion and extrinsic innervation of the olfactory epithelium. She then did a bit of a 180, and she investigated the development of the visual cortex of primates at the University of Washington. Dr. Zielinski soon came to her senses, which is a pun intended, and she returned to studying fish as a faculty member at the University of Windsor for many years. And Dr. Zielinski has now earned her stripes and is now a professor emerita. So congratulations, Barb. Barb was actually also instrumental in developing. Oh, you're welcome. The University of Windsor's prestigious Behavior, Cognition, and Neuroscience program in our Integrative Biology department. And she belongs to the Association of Chemoreception Sciences. And this association is dedicated to understanding the chemical senses. But it's here that Barb has served as a counselor. She's chaired the diversity committee and now serves on the program committee. And so without further ado, I present to you Dr. Zielinski, who will be inviting you on a journey to get to know 3 fishery chemosensory systems. Take it away, Barb. And again, we're so delighted to have you here with us today.
Thanks so much for that introduction, Tina. So I'm really, really pleased to talk to you today, to talk to all about the chemical senses. I'm going to turn my camera off for bandwidth again. So the chemical senses, and I wanted to talk about fish, but then as we got started, I thought, why don't we just focus on this great fish, the sea lamprey, a basal vertebrate. And as I'm talking about the chemosensory systems of lampreys, I will be mentioning other fish as well, too. So just the goal here is chemosensory systems, and then highlight some of our work and some of our collaborations within the last 20 or so years. Just trying to get to the next slide. Okay. Sea lamprey, here we have a nice photograph from the lab of the sea lamprey. It's a basal vertebrate. What I want you to notice, it's got a little nostril on the top. It's got these eyes, gills, and this mouth that is attaching onto a substrate. So the mouth of the sea lamprey is where its damage, where its damages some of the other species, it has this sort of jawless mouth with these teeth. In the juvenile stage, lamprey will attach itself, or it'll find some nice big juicy fish here, I guess salmonid, attach onto these fish and start feeding on them and sort of scarring these fish quite severely, and then that sort of it affects these fish and they do not last very long after this. So where are the larval stage? Is in this map I'm just showing you to show you where the lampreys spawning locations, where they're distributed. And this sort of map of the Great Lakes shows you how broadly the sea lampreys are located in the Great Lakes. These are all the spawning streams. As adults, they move into the open waters. Down here is Windsor. There are still some larvae in the St. Clair River and the Detroit River. So the Great Lakes Fishery Commission, as we heard last time from Mark Gaydon, is charged with managing the populations. And that's largely through what we call Lamprey treatment of all these streams. But what I want to talk to you about is more of the sensory world of the lamprey. So chemosensation is actually emerging of sort of neuroscience and physiology, behavior, anatomy. By chemosensation, if we think of our own bodies, we think of olfaction, of taste. And I have another system here, which I'm not sure if all you people are familiar with, the solitary chemosensory system, which we actually have. The fish have a much more developed solitary chemosensory system. We as humans, within our respiratory tracts, and it responds to various toxins. And so what I want to tell you talk today is these three chemosensory systems. Aquatic animals such as fish are submerged within this sort of chemical environment, and they are actually cueing in quite effectively to what's going on. So we're going to look at the anatomy, physiology, and all of this in the context of fisheries management. And so what our goal here is to sort of help fisheries management with their task of managing these populations, not only of lampreys, but of all aquatic animals, and then the study of ecology. So the three chemosensory systems, the olfactory system, gustatory system, and the diffuse system. So here I've written down sort of just cues for lampreys and a lot of fish too. So lampreys can smell basic amino acids, sex pheromones. The Michigan State has done a tremendous amount of work on sex pheromones. These are bile acid derivatives, and spermine also from the Lee Lab, and Scott has worked on spermine being a derivative of a basic amino acid. These are all very strong behavioral responses from these compounds that stimulate the olfactory system. The gustatory system, which is also the taste buds, we have what we call canonical taste solutions, so sweet, sour, bitter, and umami or amino acids, which we taste, is remarkably conserved in the lampreys. There was an earlier study in 1985, and we're following up on some new studies of this really interesting system. And the diffuse chemosensory system, where I'll show you the lampreys have a remarkable distribution of these solitary chemosensory cells on their exterior of the body, and they respond to not only basic amino acids, but neutral amino acids, and this really amazing response to dead trout water, which is kind of interesting. So to start with the olfactory system. So the olfactory system, which is our sense of smell, the lamprey has a single nostril on the dorsal surface, and there's like a little passageway leading to what we call the peripheral olfactory organ. So this, in our case, in our own bodies, up in our nostril, sort of close to the top of our nose, we have these olfactory sensory neurons. So backfilled, meaning we label these by injecting into the brain, and we see these are the olfactory sensory neurons in the olfactory epithelium. Cells here, and sort of this columnar cells in the epithelium. Remarkably, I think I'm using that word too much, but if we test single cells, here's an important fact that we see in the lamprey is that separate cells, these recordings are from separate neurons in the olfactory epithelium. Here we're stimulating with these are bile acids or derivatives that the Lee lab was testing. Very similar compounds. There's only some in the orientation of the hydrogen. Individual cells can pick these up, these sort of differences. And we see that time and time again where the individual cells in the epithelium can distinguish between very similar compounds. And so the lamprey behaviorally is able to distinguish various compounds that are very similar chemically, and some of that distinction occurs right at the epithelium through the binding properties, through the receptors. Now let's look at, so we're more concerned about what happens in the brain to cause movement. So lampreys will move during the migration stage. They will smell the larval pheromone, which is a bile acid derivative, and they move from the inland lakes up into the streams, or if they're in the ocean, they move from the ocean up in stream. So you have a very strong movement response. So here we have sort of a diagram of the olfactory system. The MOE is the main olfactory organ or epithelium. They have this accessory olfactory organ as well. And if you look down on the lamprey's brain, it is laid out very nice and flat. It's not all sort of curved up like the human brain. And you can see these sort of parts. So what I've labeled here is the olfactory bulb that gets projections from the olfactory epithelium, the pallium, and then going on into higher brain centers. So what we want to keep an eye on here is this area called the MLR, or the mesencephalic locomotor area in the reticulus. So this is a series of experiments. It was actually done at the University of Montreal in Réjean Dubuc's lab, where this prep for the lamprey brain was recorded from. And so you can see when into the olfactory epithelium, and the recordings are from the reticulospinal cells, which stimulate movement. Every single odorant here was stimulating a strong movement response or strong neural response to command. These are movement command neurons that will be responding to the odorant. So there's a very strong correlation between olfaction and movement. And so this sort of, after years of work, we figured out there's like a medial pathway from the olfactory bulb going right into these movement control areas of the lamprey. Even within here, as part of the things that we do as chemosensory scientists, we try to understand how the brain is interpreting odorant information. And so here we see there's an area that responds to this medial area that responds to many different odorants. So I'm just going to show you here. This is some electrophysiology. Don't have to worry. Is the most important part here is this bar graph here. So this medial region of the olfactory bulb is responding to amino acids, to cholic acids, and pheromones. It responds to all of these compounds. And that is important because that medial region is linking directly to locomotion. So it's some sort of a gating mechanism. If you can get an odorant into these cells or to stimulating these cells, you're going to stimulate movement. And it seems that it's not selecting any particular, and it's just sort of directing this movement response. The lateral part of the olfactory epithelium will only respond to amino acids. So the lateral part of the olfactory organ bulb, I'm saying, the brain only responds to amino acid. It never responds to any of the pheromones. And you'll think that's kind of weird, but the other thing that happens to this is that in other teleosts, if you look at a zebrafish, it'll be the same. The lateral part of the olfactory bulb only responds to amino acids. Maybe that has something to do with feeding. I don't know. Our own olfactory bulbs are mapped out, and there's a lot of overlap, but it also is mapped up with some odorants. So here we have amino acids that are stimulating only this lateral area, only amino acids. But if we record from this another area, the dorsal area, sort of on top, we get a lot of bile acid responses and pheromone responses, and just about 50% of the cases respond to amino acids. So there's some sort of mapping going on in the brain. Here I'm just showing more electrophysiology, more sort of responses we're getting from the olfactory bulb area. These are all the different pheromones that are stimulating movement at a different stage during migration, during reproduction, and yeah, so they're all affecting. So the other thing to think, then, as I was saying, this medial pathway goes right to locomotion. Here's the medial pathway. There's also, as we just showed, areas are also responding to odorants. They send projections to this area called the lateral pallium. The lateral pallium is a evolutionary precursor of our cortex, cerebral cortex, which we use for thinking and for integrating information. So this is a bit of an integrative center, but usually this area is pretty quiet. Yeah. What I want to look at now is, are there any circumstances in which this area that sends to integration, can it be regulated? Can it be suppressed or stimulated? So this has many modulatory neurons. It has dopaminergic, GABA, and S modulate responses. So we're recording from the olfactory bulb, we're putting on the pheromone, and usually you get, a small response from that dorsal area to a pheromone. When we applied gabazine, which will block the GABA responses, the GABA inhibition, you see there's a huge response in the olfactory bulb. So the olfactory bulb is usually relatively quiet, but when you inhibit these GABA neurons, it's going to give a huge response. So what does that have to do with the rest of the lamprey? So here we have a diagram showing that if we have under control conditions, if we record from the RS cells, these are cells that are stimulating movement, okay? Usually if you apply an odorant, you just get a small response from these RS cells, okay? But if we apply gabazine onto the medial area of the olfactory bulb, then releasing this inhibition, and you get a very strong response in the bulb, and then a very strong movement response, okay? This drug, gabazine, uses, it'll inhibit all GABA responses, even in humans, and it will stimulate convulsions, okay? But here we're just using it to see that you get, and if you sort of blow up this area, you see there's very strong movement response. VR is the ventral root, so these are motor neurons going right to muscle cells. So it's being stimulated when you release the GABA inhibition. So that's in the medial area. In the main olfactory bulb area, we have the same. So you have this big inhibition usually going on. So what we're thinking is that during migration, there might be some modulation here going on, where you might have some of this very strong movement towards the source that's being gated by the neurotransmitter. Now I want to just jump and ask, okay, what else is the olfactory used for, and how sensitive is it to things in the environment? And the answer, as from my experience, that olfaction can be pretty can be affected. If you get a cold, for example, your sense of smell is affected. And so here we have copper, which is a compound that is found in the environment sometimes, and especially if there's environmental industrial pollution. And not only in lamprey, but also in all fish, they are very sensitive to copper. So these are just levels of copper in our Windsor area. In the spring, during wash-off, they can get up to per liter of copper. And let's see what happens if you look at the olfactory epithelium, the sensory neurons in a lamprey. Even when you put like small concentrations, 5 micrograms of copper, you start seeing these olfactory sensory neurons, rather than going right to the top, they're kind of being pinched back. 10 micrograms per liter of copper. These sensory neurons, some of them seem to be sort of submerged. But what if we're recording responses from the olfactory bulb? Here's an experiment where we're giving a pheromone, 3-kpzs. We're looking at a response. And then when we applied this is recording from the brain, olfactory bulb, but when we give 10 micrograms per liter of copper, and again, give the pheromone, and you see the response is reduced. There is a bit of a recovery later when that copper is taken off. So this is like a slow, I'm just going to show you this overall for like half an hour. These are the magnitudes, they're normalized to 1. And triangles, if we applied copper and then recorded from the olfactory epithelium, this is the control up here, and so recorded from the olfactory bulb, you get a decrease very, very quickly. So there's some really quick inhibitions going on here in a copper environment. When you release the copper exposure, there's a bit of a recovery. So this just sort of to point out that the olfactory system, I'm sure, is affecting, is affected by various environmental toxins and treatments. So I've talked about the olfactory system. Now I want to jump into the diffuse chemosensory system. So this system in lampreys is located on gills, on nostril and gill pores. It's connected again to the brain. At these various locations. We have these little papillar structures on the gills, and if we look at, this is Tina Suntrace's work here at Clear, and we see that if we look at these little gill pore papillae and just go really, really close, you can see these little microvillar cells. This is a fracture. And these cells on the papillae are innervated. But what's remarkable is that if we look at the different stages, the parasite stage, when they're juvenile, you have these papillae they're present but not very well developed. But during the spawning phase, these papillae are really well developed. We have lots of these microvillar cells, and then she did some counts, and here juvenile, which is the parasitic phase. Here we have the migrating adults and the spawning adults, and the number of SCCs per region increases quite a bit. And so these fish during this stage, these lampreys, are not feeding, but they still have their olfactory systems, but they're still sensing the chemical environment. So here's, again, electrophysiology, recording from these, listening to the neurons, and these are and this is the spike integration. And we see, there are stimulants of this, what we call dead trout water is really strong. So this was discovered by Bottrop and Joving back in the 80s. They took a dead trout and put it in the water and applied, and they got a strong response. And we tried also bile acids too. So the trout liver will produce bile acids, and these bile acids are strong as well. So what we did in conjunction with Weiming Lee's lab was to take a bunch of dead trout water and analyze for what was in it. So this is what we found. This are quite a bit of bile acids. So this is Kay Lee and Weiming Lee's lab, and they found this taurocolic acid, which then lampreys can also smell, but it stimulates this SCC area. And when they ran this through PLC and mass spec, they found that there was a 498 molecular weight compound, which they hypothesized could be one of these bile acids. So these bile acids are all pretty well the same, but they have a group at a different location. And so what Assam Mohammed and Gianfranco Grande did was they tested all of these compounds on those papillae and wanted to see which one was stimulatory. So here this summarizes some of their information. So here we're looking at the top. The top one here is the parasitic phase. This bottom panel is the spawner phase. And if we look at the parasitic phase, and we're looking at different concentrations of the bile acid. So the blue bar is taurocolic acid, the green bar is TDCA, taurocolic acid. So all these THC, these are all very similar compounds, but we see that this blue one being TCA and the green one being TDCA, at there's lower concentrations, it seems to be. Okay. Once we get to the higher, these are relatively high concentrations, 1 millimolar compared to the olfactory system, which even micromolar or nanomolar will stimulate. But we do see that they are more at lower concentrations. These taurocolic acid and TDCA seems to be stimulatory. all the different locations, the nasal pore for those little papillae and the fins, and they're all responding, with neuron with action potentials. So I'm going to move now to the third chemosensory system, and that's the gustatory, or the taste system. And so for us, we stimulate, or we respond to taste, sweet, sour, bitter, and umami, which is like an MSG or like a chickeny flavor. But let's see what we see in the lampreys. So lampreys do not have taste buds on their mouth, okay? They just don't. They have teeth on their mouth, but then if you go beyond their mouth, there's a structure called the pharynx. So the food that's consumed goes into the pharynx, but the pharynx is also connected to the gills, okay? So there's like gill pores that are connecting to the external environment. So this pharyngeal area, this pharynx, is where the taste buds are located, okay? This is like a dissection showing you we have this gill pore, internal gill pores, and the pharyngeal wall. So in that lamprey has this area, we have nerves, you have the cranial nerves, specific cranial nerves coming in here too, similar ones, I think it's 9 and 10 that we have in humans, so it's innervated. If we want to know where are the taste buds, so here we have Zinat Arangzaman. She did some serial sections through juvenile and adult salmon, and she found these taste buds that are labeling just like we see taste buds labeling in mammals as well, remarkably similar with innervating fibers, taste cells, and they are on these little taste bud areas on the lateral surface of the pharynx between the gills. And here, Hassan Polat and Sharon Lackey at Gleer looked at this area of the pharynx, and you can see these taste buds with this collection of taste cells, very similar to what you'd see even in a human, this sort of taste buds. To respond to, and again, we're right now looking at regionality in this area, but I'm going to give you some preliminary results. So sucrose, they can taste sweet, okay? Denatonium is bitter, and it's usually a compound. If something's bitter, then the animal will move away from it. Proline is not really a stimulant. This is the control ringers. Okay, so they do have this really sensitive taste system, but it's not in their tongue and it's in their pharynx. Okay, so I just want to summarize what I've discussed about today. And if we look at other fish, teleosts also, they have taste buds, such as catfish or other, even zebrafish, but a lot of their taste buds are on exterior of the body as well as in the pharyngeal area. So what have we sort of learned? We know there's anatomically distinct main and accessory olfactory subsystems, different bulbar mapping, importantly, very strong connectivity, neural connectivity to motor control centers, modulation of the olfactory motor response by various neurotransmitters, including GABA. The SCCs, this area that's on the fins, on the exterior cuticular surface, is very sensitive to bile acids. Why is it responding to these trout bile acids? We don't know. That's got to be sort of try to understand that. And taste buds also are very sensitive to canonical taste stunts and are probably affecting lampreys. If it can't taste its fish that it's eating, it's probably not going to stick on. So it's probably has very strong, and that's another thing we're looking at, connections to the movement, so that it'll stay attached, I guess, when it's tasting something. Thank my collaborators, the many faculty members, Xiuming Zhang, who's in biological sciences in University of Windsor, Gianfranco Grande, I'm not going to name everybody, but these are really, really great people who I've worked with at the University of Montreal, Réjean Dubuc, Raylan Dacfus, Francois Auclair, and at Michigan State University, Wei Ming Li and Kay Lee, being very helpful. And of course, USGS Hammond Bay, Nick Johnson and Scott Meals for being really great soundboards and helping us with getting the lampreys. So thank you.